Hey everyone, if you're gearing up for the CSIR Net Life Sciences exam, you already know that cell biology is an absolute maze of intricate pathways. But today, we're zeroing in on an organelle that, honestly, gets a bad rap. It's way too often dismissed as just the cell's basic garbage bin. But as you're going to see in this explainer, the lysosome is actually a highly regulated, incredibly precise recycling facility. Truly understanding its mechanics, its delivery routes, and how it traffics its enzymes is absolutely crucial for acing your exams. So let's get into it.
Okay, let's dive right in. Here's our roadmap for today. First, we'll explore the cell recycling center. Second, we'll map out the pathways to the lysosome. Third, we'll break down the autophagy process. Fourth, we'll look at trafficking lysosomal enzymes. And finally, we'll wrap up with lysosomal storage diseases. All right, Section 1, the Cell Recycling Center, Structure and Function. So what actually is a lysosome? At its core, it's a membrane-enclosed organelle that acts as the cell's primary digestive hub. Inside, it's packed with about 40 different soluble hydrolytic enzymes. In biology, we usually refer to these as acid hydrolases. This specialized workforce includes nucleases to cleave nucleic acids, proteases to break down proteins, lipases for lipids, plus a whole bunch of others like phosphatidylsetases and sulfatidylsetases. Working together, they have the power to break down virtually any macromolecule the cell needs to clear out. And what's really interesting here is the number 5.0. That's the highly acidic pH maintained inside the lysosome, and it's absolutely necessary for those acid hydrolyses to do their jobs. But how in the world does it stay so acidic? Well, it's driven by V-class H plus ATPases that are constantly pumping protons inside. But here's the catch. These proton pumps literally cannot work alone. Think about it. If they just pumped positive protons in, they'd quickly create this massive charge gradient and completely stall out. So they strictly require the presence of anion or cation transporters to balance out the electrical charge. This allows for continuous acidification without totally throwing off the cell's electrical balance. And the lysosome isn't just some static bubble either. It goes through a really dynamic maturation cycle. It actually kicks off when an immature, late endosome fuses with an existing lysosome. This fusion creates this sort of hybrid structure called an endolysosome, and that is where the active digestion really begins. Over time, as all those materials are broken down, this endolysosome matures and converts back into a classic hydrolytically active mature lysosome. And then it's ready to fuse with the next incoming late endosome. It's a beautifully continuous cyclical process. Moving on to Section 2, Pathways to the Lysosome, Delivery Routes. Think of this recycling facility as having 4 main loading docks. First is phagocytosis, which the cell uses to gulp down really large particles, like invading pathogens or bacteria. Second, we have endocytosis, which is usually receptor-mediated to bring in specific extracellular molecules. Third is macropinocytosis. This is a much more non-specific uptake, basically taking big gulps of extracellular fluids and solutes. And finally, autophagy, which is the cell's internal mechanism for breaking down its own damaged organelles or cytosolic bits. 4 totally unique roads, but they all ultimately lead to the exact same destination, the lysosome. Now here's an incredibly crucial insider tip for your exams regarding macropinocytosis. This specific pathway is actively stimulated by the oncogene RAS. You see, in normal cells, proto-oncogenes just promote your standard healthy cell division. But if a gain-of-function mutation happens, they flip into oncogenes. Cancer cells very frequently have a constitutively active form of this RAS protein. Because it's permanently switched on, this RAS aggressively ramps up macropinocytosis. It literally allows the cancer cell to constantly drink up surrounding nutrients to fuel its own out-of-control growth. Let's look a bit closer at receptor-mediated endocytosis. The golden rule here is that unliganded receptors are practically never endocytosed. The whole process only starts when a specific ligand binds. Once it does, a ubiquitin tag gets slapped onto the receptor, marking it for degradation. In the early endosome, these specific escort proteins recognize those ubiquitin tags, which causes the membrane to fold inward, pulling the receptors inside. This creates what we call a multivesicular body, or late endosome, which finally fuses with the lysosome, effectively hitting the off switch on the cell signal. Which brings us perfectly to Section 3, the autophagy process, self-eating. Autophagy doesn't just happen randomly. It's under very strict management. And the big boss here is the mTOR kinase. As long as the cell has plenty of amino acids and isn't stressed out, mTOR stays highly active. And a highly active mTOR strictly blocks autophagy. It's only when the cell is facing real trouble, like starvation, hypoxia, or severe damage, that mTOR gets inhibited. This essentially lifts the brake pedal, allowing the cell's self-eating recycling process to finally kick into gear. When autophagy does trigger, it goes through 4 distinct steps. It starts with nucleation, which is sparked by the ATG1 complex producing PI3P. That signals the extension phase, where ATG8 handles homotypic fusion and ATG9 handles heterotypic fusion. Together, they expand the membrane into this sort of cup-shaped structure called a phagophore. Next up is closure, where those edges seal shut to form a complete autophagosome. And finally, snare proteins step in to mediate its fusion with the lysosome so the actual degradation can happen.
Now pay close attention to these hybrid terms because they pop up on exams all the time. When an autophagosome fuses with a lysosome, we call it an autolysosome. If it happens to fuse first with a late endosome, that's an amphisome. And as we mentioned earlier, a late endosome fusing with a lysosome creates an endolysosome. Lock those 3 terms into your memory, they're easy marks.
But that raises a really fascinating puzzle, right? If the lysosome is absolutely packed with these aggressive proteases meant to shred proteins apart, how do the cell's freshly made, fragile digestive enzymes survive the trip there in the first place? How do they not just digest the cell along the way or get completely destroyed themselves? Let's answer that in Section 4, Trafficking Lysosomal Enzymes, the Workers. The journey these soluble enzymes take is quite the trek. First, they're synthesized in the cytosol. Then they're imported into the endoplasmic reticulum lumen, where they fold up and get a base N-linked oligosaccharide attached. After that, they get packed into CO2-coated vesicles and shipped right over to the cis Golgi. It's actually here, in the cis Golgi, that they receive their incredibly specialized targeting tags. Once tagged, they travel across the Golgi to the trans-Golgi network, get packaged up into clathrin-coated vesicles, and eventually make their way down to the endosome and finally, the lysosome. That specialized targeting tag I just mentioned, that's known as mannose-6-phosphate, or M6P. You can think of this as literally writing out the exact postal address for the lysosome. And it happens in 2 very deliberate steps, using 2 resident cis-Golgi enzymes. In step 1, glic-ANAC phosphotransferase binds to a signal patch on our hydrolase and transfers a glic-ANAC phosphate directly onto the 6th position of a mannose sugar. Then, in step 2, a second enzyme called phosphodiesterase comes along. It acts a bit like a pair of scissors, snipping off the glic-ANAC portion, but crucially leaving the phosphate right there. The result, the M6P address tag is finalized, completely readable, and ready to go. So the really crucial point here is how the cell uses pH to pick up and drop off these enzymes with perfect timing. Over in the trans-Golgi network, the TGN, the pH hovers right around 6.5 to 6.7. At this specific pH, the newly tagged enzymes bind super tightly to M6P receptors, which allows them to get safely loaded into those clathrin-coated vesicles. But watch what happens next. Once that vesicle fuses with the late endosome, the environment shifts to a much more acidic pH, dropping below 6.0. That sudden drop in pH physically alters the receptor, forcing it to instantly release the enzyme into the endosome. The enzymes then safely move on to the lysosome, while the empty M6P receptors are packed into retromer-coated vesicles and shuttled right back to the Golgi to be used all over again. It is a brilliantly elegant pH-dependent shuttle system. Finally, let's look at Section 5, Lysosomal Storage Diseases, when the system breaks. Because what actually happens when this incredibly elegant system breaks down? Well, for your exams, you really need to grasp the key difference between these 2 conditions. In Hurler's disease, the problem is a mutation that affects just one single individual lysosomal enzyme. It's bad, but it's localized. But I-cell disease, that is systemic and incredibly severe. In I-cell disease, the patient is completely missing glic-NAC phosphotransferase, that vital first cis-Golgi enzyme we talked about. Without it, absolutely none of the lysosomal enzymes get their M6P postal address. The entire workforce gets missorted and secreted right out of the cell, leaving the lysosomes completely empty of any digestive power. It really puts everything into perspective, doesn't it? Just think about it. When a single missing molecular shipping tag, literally just a tiny phosphate group, can cause an entire cellular recycling system to collapse and leave the cell choking on its own waste, It truly highlights how much of our macro-level, everyday human health depends on these invisible, remarkably precise microscopic postmen just doing their jobs perfectly every single second of our lives. I really hope this explainer shed some light on the brilliant mechanics of the lysosome for you. Thank you so much for joining me. Keep studying hard, and I will see you in the next one.